I had pizza last night and then I instantly had heartburn. But I bit into the pizza and I instantly burned the roof of my mouth and the inside of my lower lip, right? Of course. And in doing so, I killed some of those skin cells of my lip and my cheek and the roof of my mouth. And now there are cells that need repair, but there's also cells that just need replacing. Part of that repair will involve replacing those damaged cells. Those replacement cells, they're going to be provided by copying some of the nearby undamaged cells. Luckily, this is a small wound and it won't take long to heal it, but it still involves producing thousands of new cells through a process called cellular reproduction, which can also be called cell division. At my age, the principal function of cell division is the repair and replacement of damaged cells. But another function of cell division is growth. You are built from approximately 60 trillion cells, and every single one of those cells can trace its origin back to the single fertilized egg that formed about 9 months before you were born. So every cell in your body today was formed by way of cellular division, starting from that first cell. Cell division ensures that the set of genetic instructions passed on to each of your 60 trillion cells is complete and hopefully identical to that first cell. Okay, so different kinds of cells divide at different rates depending on their role. There are some human cells, like your skin cells and the lining of your digestive tract, that divide every day. But then there's others, like nerve cells, that are so specialized they never divide at all. And this may help explain why brain injuries and nerve injuries, they never heal. Well, there's a low probability. Cells that do divide, they follow a sequence of steps known as the cell cycle. What I have pictured here for you is a stem cell, and it does have a purpose that it will be a neural cell in the end, but this stem cell is capable of dividing into any of these 3 cells that you see here, which are all cells called neuroglia. And neuroglia are cells that they're not cells that conduct action potentials, but they're kind of like housekeeper cells for neurons. This slide reinforces how important it is to have a background on DNA and the cell cycle before moving forward with mitosis. So there are some mini lectures. Recall that the cell cycle has, it really has 2 phases. It really just has interphase and then a cell divisional phase that is either mitosis or meiosis, but this right here is interphase, consisting of its 3 subphases, G1, S, and G2. When a cell enters into mitosis, it's already been through all of these 3 subphases. It's required that chromosomes are in their duplicated state with their sister chromatids attached at the centromere. This duplication is what occurs in the S phase, so named because it refers to the synthesis of new DNA, or the synthesis of the sister chromatids. Because we are approaching a cell division phase, it also means that the chromosomes will be condensed enough to be seen with a light microscope. Remember, in interphase, you can't see the chromosomes. They're being duplicated, or they're being used to make proteins for growth. Along the bottom of this slide, I have 3 cells. The first cell has 4 chromosomes in an unduplicated state, just like strings. The middle cell has 4 chromosomes. They are in their duplicated state with their sister chromatids attached, but they're uncondensed, so they'd be in like G2. This last cell also has 4 chromosomes, and they're in their duplicated state, condensed, and ready for mitosis. The phases of mitosis can be split into 4 or 5 phases depending on who you ask. I recognize 4. It's just easier. And let's list these 4 from left to right here. This is prophase. This is metaphase. This is anaphase. And this is telophase. And we'll discuss what the last one is in just a moment. To tell these pictures apart here, there are a few things that you can look at. You can look at the size and the shape of the cell, the extent of the green mitotic spindle, and the placement of the blue and red chromosomes. So when a cell approaches mitosis with its chromosomes in condensed state, it enters prophase. In prophase, the nuclear envelope disappears, which is why you see a dotted line in the first picture here. And the centromeres, or the centrioles, move to opposite sides of the cell, and the mitotic spindle starts growing some fibers from them. In the second picture, you can see that the most defining characteristic of metaphase is that the chromosomes are lined up in this orientation along an imaginary line called the metaphase plate. If the alignment is incorrect, if it's not like straight down the middle of the chromosomes, it could result in 2 cells that are not identical copies of each other, and they're not identical copies of the original. The third phase here is called anaphase, and this phase is actually quite energy intensive, even though sometimes it can look like nothing is happening when you view it under the microscope. Anaphase is basically a tug of war. And that's what's happening, is that the spindle fibers that are attached to the centromeres, they start to pull in opposite directions, break the centromere apart, and pull the sister chromatids to the opposite ends of the cell, right? Kind of like taking 2 pieces of paper that are stapled mistakenly, ripping them apart, and me giving 1 copy to one student and then the other copy to the other student. Man, that would happen so much when I used paper. The last phase, which is called telophase, is basically, it's just the opposite of prophase with a little bit more. The chromosomes become dispersed. The mitotic spindle starts to disappear. The nuclear envelope begins to reform, noted by the dotted lines. And this structure here between the 2 potential cells becomes developed. In an animal cell, this structure between the 2 cells is called a cleavage furrow. And actually, I just want to label it for a moment, even though my pen is not really working. But sorry. It is this structure here that is the cleavage furrow. So the cleavage furrow, it's going to separate these 2 cells entirely in a process called cytokinesis. Many students wrongly think that telophase and cytokinesis are the same phase. They're not. Now, is cytokinesis part of mitosis? Again, it depends on who you ask. Some books include it, some books don't. Telophase, it results in the complete separation of the genetic material that's potentially within the nuclear envelope, right? So telophase is concerned about the nucleus. Cytokinesis is the separation of the contents of each cell, so all the stuff that's not in the nucleus. Just as there are checkpoints in the cell cycle itself, there are checkpoints that are very specific to the phases of mitosis. And this diagram here kind of shows you some of the most common issues with the metaphase plate and with the mitotic spindle, which are the 2 most common things. A simple little thing like 1 extra centriole in your centrosomes can result in this picture up top here that says multipolar spindles. A cell encountering multipolar spindles would be forced into apoptosis or cell suicide before it could complete mitosis. There are proteins that can detect these cell division errors, and those proteins can turn on the genes for apoptosis. Although there are checkpoints, there are really some common mistakes that consistently happen in mitosis, and these mistakes even happen in meiosis. Most of them have to do with alignment on the metaphase plate, and these mistakes are called non-disjunctional mistakes. Try to think of the resulting two cells from each of these mitosis mistakes that you see here. I'm going to encourage you to draw the resulting two cells and see how they are not the same. Basically, these types of mistakes here, they don't result in the cells being clones of each other. And these types of non-dysfunctional mistakes usually result in one cell having more or less genetic material than the other cell. Cancer is an overproduction of cells resulting in a tumor. This would be the cell cycle completely out of control, not only doing too much mitosis, but also allowing cells that shouldn't do mitosis to do it. So cells that are not meeting the checkpoint criteria, they're not being removed before they have the chance to complete mitosis. This results in a cancerous or a precancerous cell, making many copies of itself and resulting in a tumor. The tumor cells will be misshapen. They'll not serve their intended purpose. And as these mutated cells continue to divide unregulated, they'll amass into a tumor. If the tumor becomes large enough, number one, it'll get its own blood supply, but also fragments of the tumor will start to split from the original mass. They'll enter the bloodstream and they'll become deposited in other tissues, most commonly the spine and the lungs at first. And this is what metastasis is. Most cancer drugs target some aspect of mitosis and they prevent it from happening, right? For example, many cancer drugs prevent the mitotic spindle from forming. They prevent the nuclear envelope from breaking down in prophase, or they prevent the cleavage furrow from performing cytokinesis. But in all of these instances, the cells that are trying to divide are then going to be randomly removed by apoptosis. The problem with cancer drugs is that many of them are systemic, meaning they affect all the tissues in your body, not just the dividing tumor. So this means that while under the treatment of these drugs, new cells in the mouth, new cells in the digestive system, new cells on any epithelial tissue, such as the lining of your bladder, those cells will not form. Therefore, systemic cancer drugs prevent all acts of mitosis in the body, kind of leaving the patient open to immune system intrusion because of the lack of making white blood cells. There are other cancer methods of treatment, one being surgery and the other being radiation. These methods are not systemic, but have just as long a list of cons as any of the more cancerous systemic drugs.